segment, questions related to prior knowledge. Now come to the topic, and today's topic is recombinant DNA technology. So what are the steps involved in recombinant DNA technology? So very first step is isolation of DNA. And then we are going to fragment the DNA, because DNA is a long thread, okay, and we can't identify the sequence of DNA in a long thread. So we have to chop the DNA. And for chopping the DNA, we have molecular scissors. Can you name the molecular scissors? Yes, it is mentioned over here. Can you read out? Molecular scissors. Restriction endonucleases or yes, restriction. Okay, I got your answer. So restriction endonuclease, these are molecular scissors. They inspect the whole length of the DNA, okay, and they recognize their recognition sequence and then make a cut in the DNA so that DNA gets fragmented. Okay, now all the fragments are mixed in a test tube. Now by the help of gel electrophoresis, okay, we are going to separate DNA fragments. Got it? Now by the help of gel electrophoresis, when the DNA fragments get separated, we are going to isolate DNA. So isolation of DNA will be easy when the DNA fragments get separated from each other. Now we know that this fragment has our desired gene. Now by the help of PCR, what is it? Who is going to give me the full form of PCR? Yes?
Polymerase chemical reaction.
Polymerase chain reaction. Very good. Polymerase chain reaction. So by the help of polymerase chain reaction, we can amplify the gene of interest. Okay, the gene of interest is foreign gene, alien gene, the desired gene. Say insulin is the desired gene. Okay, so we are going to make multiple copies, billions of copies of desired gene, which we call amplification. We can amplify, we can make multiple copies by the help of this machine. We are going to study this also in detail. All the points we are going to study in detail, but let us look all the steps which are involved in recombinant DNA technology. Now after getting the desired gene, ligate it. Ligate means just paste it to the vector. And what is the vector generally we used? Plasmids. Okay, and can you tell me the name of the enzyme which helps in ligating the foreign DNA, which we call molecular glue? What is it?
DNA ligase.
Very good. It is mentioned over here, DNA ligase. So DNA ligase is the glue which is going to ligate, which is going to join your gene of interest, your desired gene, your foreign gene, whose product you need, okay, to the vehicle DNA, or you can say the vector DNA. Here the vector we are taking plasmid, okay. Now, after taking foreign DNA, the plasmid becomes recombinant DNA. What is recombinant DNA? A DNA that has foreign gene. And you are going to transfer that recombinant DNA into the host. And host may be a bacteria, host may be a plant cell, host may be an animal cell. It depends. Okay. Now, culture that host on large scale, on commercial scale in a bioreactor. So what is a bioreactor? Bioreactor is thought to be a huge vessel in which we can culture approximately 1000 liters volume of bacteria or any animal cell which has our desired gene. So volume may be more than 1000 liters, okay, so that we can get our desired product. For example, our host cell has insulin gene, so insulin gene can be extracted on the commercial scale by the help of bioreactor when we culture our host cell with desired gene in a bioreactor. And afterwards, what is the last step? Downstream processing. What is it? We get the product. We withdraw our desired gene from bioreactor. Now, our desired gene our desired gene product, which we call recombinant protein, has some impurities. So these impurities has to be removed, and by removing, by separation, you can say by purification, okay, of our desired gene product more specifically, then we call it downstream processing. For example, say insulin. So we are going to isolate insulin from the bioreactor. So it has lot much of impurities. So by the help of downstream processing, we can purify, we can isolate our desired desired gene product, which we call recombinant protein, which may be a therapeutic drug, okay, in purified form. Got it? Now come to the very first step. We are going to study the very first step of isolation of DNA. Okay, before that, let us have a look of all these points by the help of diagram. Let us say this is the diagrammatic representation. Here you can see what is the first step of recombinant DNA technology, bacho? What is the very first step?
Isolation of DNA.
Very good.
Isolation of DNA.
Okay, I got the answer. Very good. So isolation of DNA is the very first step. So here is the human cell. Here is the bacterial cell. So from both the cells, we are going to isolate the DNA, and how that we already have done in practical. And today also I'm going to explain it in detail. Okay. So here you can see from human cell, from the nucleus of human cell, we isolate DNA, and same with from the bacterial cell, we have to isolate the DNA. This is very first step. Now, because DNA is a long thread, it is in, if you talk about human cell, it is 2.2 meters long. And if you talk about bacterial cell, DNA, it is 1.33 mm. So it is very long. So any long, ultra thin thread can't remain straight for long. It gets coiled up. Okay. So just to avoid coiling, because we need genes, so we chop them, we cut them. And for cutting, we need what restriction endonuclease. Okay, here we used EcoR1. Okay, you know the nomenclature. Okay, because it is already explained. We know the nomenclature of EcoR1. E for Escherichia, Co for coli, R for the stain, and first is the Roman numeral is what, in which order the enzyme is isolated from E. coli bacteria. And what is the role of this EcoR1 enzyme? It is actually a molecular scissors. It inspects the whole length of DNA, recognize its recognition sequence, and make a cut in the DNA. And we use same enzyme in both the cases, in human DNA as well as in case of bacterial DNA, which is actually a plasmid. And you know the plasmid is extra chromosomal DNA. Okay, so here you can see this is bacterial DNA. So along with bacterial DNA, some bacteria have extra chromosomal DNA, which is circular, and they are autonomously replicating. They replicate by their own. They are not dependent on bacterial chromosome. Okay, they have their own ori. They can replicate by their own. You can make a cut in them. You can remove the genes except ori. You can introduce foreign gene. Still, they do not, stop multiplying, autonomously replicating. Okay, this is the unique property of plasmid, as well as they are the vehicles of the gene, of the desired gene. They carry gene from one organism to another organism. Got it? So this is why we select plasmids, just to transfer human gene into the bacteria. Got it? Now here you can see we cut the DNA by EcoR1. And can you tell me the recognition site of EcoR1? Any one of you? Can you tell me the recognition site of EcoR1? You have not learnt it?
GAATTC.
Very good. GAATTC. Okay. So this is what is GAATTC 5 prime. Here you can see 5 prime to 3 prime. Got it? 5 prime to 3 prime. Similarly, here you can see because the two strands of DNA are what? Complementary to each other, and they are anti-parallel to each other. So here you can see 3. Here you can see 3 prime. So what pairs with G? What pairs with G? What pairs with G? C. Very good. Very good. And what pairs with A?
Z.
What pairs with A?
T.
Very good. T.
T.
Very good. And what pairs again with T? Okay. And what pairs with T?
A.
Very good. Okay. And what
A.
pairs? Very good. What pairs with uh, C? It's G. And what is the polarity?
G.
Very good. And what is the polarity?
5
5 prime.
prime.
Yeah, your mic is not working properly, bache. It's your voice is cracking actually. Okay. So but you participate. Okay, now here you can see this is what is the recognition site of eco R1 GAATTC from 5 prime to C prime, and from 5 prime to C prime in this direction GAATTC. Such sequences are known as palindromic sequences, and eco R1 has to recognize these sequences and make a cut between G and A. Where is the cut made? Between G and A. Here you can see. Okay, if I make a cut, here you can see. So I'm going to make a cut between G and A, sugar phosphate. Here you can see. Okay, so I'm sorry, I have to clear it. Let me draw it again. Let me draw it again. Yes. So here you can see, this is the cut. Wait a minute. This is the cut. Here we need the cut. I'll show you with the help of marker. Let me show you with the help of marker. From here we are going to cut between G and A. Okay. So if I make it again, so I'm going to make it again. Yeah. From G and A. Here you can see between G and A. So here is the cut made. Here you can see. So this portion, I'm going to shade it. This portion is going to remove from the DNA. Here you can see. Okay, only from AATTC and here from G. So what is left? G, C, okay, and TTA. So TTA remains free. It is single stranded. Here you can see it remains free. Okay, here you can see it remains free. So these free single stranded ends are called sticky ends. That is why we cut the DNA with the same enzyme. EcoR1. Same enzyme is used so that we can get sticky ends. And these sticky ends are complementary to each other. If, for example, here is AATTC. So what should be here? Hello, mute yourself. What should be here? What should be here? If here is AATTC, so what should be here? Speak up. Here should be what? Yes, speak up, bache. If here is AATTC, so what should be here? What is the complementary of
G.
TTAAG? Okay,
G.
so yeah, that is why we cut the two DNAs by same enzyme so that we get the sticky ends. And this DNA fragment, let us say this has human growth hormone gene. Okay, and we are going to easily ligate. Ligate means we can easily stick, we can easily join our gene of interest, our desired gene, our foreign gene, okay, into the plasmid DNA. Got it? Now, after taking foreign gene, this plasmid DNA becomes the recombinant DNA. This is recombinant DNA because this DNA doesn't have growth hormone gene. Okay, we introduce it from outside, from human chromosome. From human DNA, we isolate the growth hormone gene and we introduce it by the help of which enzyme? What is the name of the enzyme which is going to ligate? Yes, pick up. Manvi, you tell me, bache. Manvi, give me the answer. Manvi.
Ligase.
Very good. Why can't you speak, bache? Participate, please. DNA ligase. So DNA ligase is the enzyme which is going to join our desired DNA fragment, which has our desired gene, into the foreign DNA. Or you can say in the vehicular DNA or a plasmid. Now, this plasmid, this DNA becomes what? Recombinant DNA. Now introduce this DNA in the host. Here host is bacteria. Now culture this host on the large scale and what do we get? Our desired product. And here what is the desired product? Om Bhutani, what is the desired product over here? What is the desired product?
Growth hormone.
Very good. Growth hormone is the desired product. Human growth hormone. So by the help of bacteria, we are going to get human growth hormone or human hormone we are going to get. Got it? Now students, come to next. Yes, Harshit, you raised your hands. If you have any problem, please ask. I'm going to change the slide. Okay. Now, okay, no problem. Okay. Now come to next. Yes, Om Bhutani, you raised your hands, bache. Yes, Om, you have any problem, please ask. Okay. Okay, students. Now, students, here you can see how genes, how DNA, you can say, isolated from from the different plant sources, different animal sources, different, you can say organisms. Here you can see, if you are going to, hello, better mute yourself. If you have no queries, please mute yourself. Or you raise your hands if you have a query. Mute yourself, please. Okay. Mute yourself, bache. There is a chatting box. You write your query over there. I read, I go through it, and I let you know what is your problem. Okay. Now, see, you can isolate DNA from bacterial cell, plant cell, fungal cell, but all these cells have cell wall. So to reach up to DNA, you are supposed to digest the cell wall. Okay. So because all these first three organisms have different chemical components of cell wall, so different enzymatic reactions are required. For example, bacterial cell wall is made up of peptidoglycan, which can be only digested by lysozyme enzyme. Plant cell wall has cellulose and pectic substances. So respective enzymes are required like cellulase and pectinase. Fungal, if you talk about fungus cell wall, so fungus cell wall is made up of chitin. So respective enzyme chitinase is required. Now we digest the cell wall. Now what is left? Plasma membrane. And you know that plasma membrane is made up of lipids and proteins. Similarly, nuclear membrane, which is made up of what? Lipids and proteins. Got it? So bacterial cells don't have a nuclear membrane, but yes, rest of the cells have nuclear membrane. So we are supposed to digest proteins as well as lipids. But bacterial cell has a cell membrane. So they do have bacteria. I mean to say they do have proteins and lipids. So we need respective digestive enzymes to digest proteins and lipids. So here you can see, to digest proteins we need proteases enzyme, to digest lipids we need lipases enzyme, to digest RNA we have ribonucleases enzyme. After chemical treatment, after enzymatic treatment, only one macromolecule is left in the content. And what is it? What is it? What is left after digestion of all the macromolecules? Though lipid is not a macromolecule, but
DNA.
very good. Only DNA is left. Is it Mirdul? Mirdul gave the answer?
Yes, ma'am.
Okay, very good. So here you
Yes,
can,
ma'am.
okay, beta, thank you. So here you can see, bache, that after adding chill ethanol, okay, because DNA is soluble in water, but it is insoluble in ethanol. So when we add chill ethanol, your DNA is going to precipitate out just like a cottony thread. Okay, and you can spool out. What is the meaning of spooling? Spooling means these fine threads of DNA can be wrapped around the glass rod. That is what is called spooling. Got it? So in this way, we can isolate the DNA from any cell, maybe bacterial cell, maybe plant cell, maybe fungal cell, maybe animal cell. Because animal cells don't have cell wall. So these treatments are not required. But yes, we are supposed to dissolve their RNAs, we are supposed to dissolve proteins, lipids, except DNA, we are supposed to dissolve all the, or digest, you can say more accurately, all the major components, right? Now come to the next point. Here you can see, students. Now, let us change the slide. Is the slide changed? Have you got the new slide? Hello.
Yes, ma'am.
Okay. Now, students, here you can see this is NCERT description.
Yes, ma'am.
Yeah, and this comes in 3 marks in your board examination. This we already discussed, how the vector DNA, which is plasmid DNA, and the foreign DNA, say human insulin gene, we are going to isolate from human DNA. So we are supposed to cut by the same restriction enzyme. Here you can see how GATTC is being cut between G and A in the sugar phosphate bond. Okay, and you can get the sticky ends. Here you can get the sticky ends. What are sticky ends? These are free ends of DNA which are single stranded and complementary to both the DNA's vector and the foreign DNA, so that they can be easily ligated by the help of enzyme DNA ligase, and in this way we can get a recombinant DNA. This is how recombinant DNA is formed. Clear? Now students, come to next. Here you can see, come to the next slide. Separation and isolation of DNA fragments. So because you chopped your DNA,
Ma'am, next slide.
you haven't got the next slide. The next slide is not visible. Is it visible or not?
मैम आ गया yes ma'am it's visible
Okay, so because you chopped your DNA, and why you chopped your DNA, you are very well aware, because your DNA is very long thread, and you can't isolate that long thread, DNA. You can't isolate desired gene from that long thread DNA because it gets coiled up. So just to isolate a particular gene or desired gene, you have to chop the DNA, right? And because when we chop the DNA, all the fragments, small, large, all are mingled up, all are mixed up in a test tube. Now, there is a technique to separate all the fragments from each other. Okay, and this technique is called gel electrophoresis. Okay, and here you can see, students, how by the help of gel electrophoresis you can separate what your DNA fragments. So there are two principles behind gel electrophoretic machine, behind separating DNA fragments. Very first principle, you can say, is that DNA fragments, or simply DNA, are negatively charged, right? So when we provide electrodes, positive and negative electrodes, you know that negatively charged molecules move towards positively charged electrode, that is anode over here. Okay, and another one is, here we use another principle lying behind gel electrophoretic machine, is that here we use agarose gel, which we derived from seaweeds, agar and agar, okay, that we get from red algae, gelidium and gracilaria. And these fragments are going to be separated by the help of their size, because agar agar is available as powdery form. Mix it by the help of buffer solution and using some electrolytes, mix it in water, having electrolytes that we call buffer solution to maintain a particular pH, provide a particular temperature, for example 40 degrees Celsius, and this powdery form is converted into paste, and then after providing a suitable condition, it solidify, and when it solidify, it forms a mesh like structure. Here you can see, if I can draw the diagram, here you can see it forms a mesh like structure. Here you can see in this form, and when a mesh like structure is formed, so some pores are created. Here you can see some pores are created. Here you can see these are the pores. Okay, and through these pores, as per the size of DNA fragments, the DNA fragments move from cathode to anode. So you can say that agarose gel provides sieving effect of the DNA fragments. It gives sieving effect. Chalne ka kaam karta hai. It gives sieving effect. It sieve the molecules. It helps in resolving or separating the DNA fragments. So smaller molecules move forward and the larger molecules remain behind. So this is the principle behind gel electrophoretic machine that we did already, right? Now students, come to next. Got the point? Got this point? Have you got these two? Yeah,
Yes, ma'am.
these two important principles behind gel electrophoresis. Okay, now students, come to next slide. Now here you can see, this is the electrophoretic machine. Here you can see negative and positive terminals. These are very much visible. Here you can see, okay, and Here you can see, here is the buffer solution. Buffer solution has electrolytes. So because electrolytes are must just to carry electricity. Okay, so electricity can only, or electrons can only move when there is electrolyte. Okay, and here is the gel plate. Here you can see, and here how the DNA fragments are separated. Okay, so this is what is gel electrophoretic machine. So two principles are behind: DNA being negatively charged, move towards positively charged anode, number one, and number two, DNA fragments are resolved on the basis of their size. Got it? Now students, there are some questions that we call post assignment. Okay, because we have done these two topics, so let us talk about, because time, I have got the warning by the Zoom, only 5 minutes are left. So what are the two important tools that give birth to genetic engineering? Yes,
Both
yes,
of them.
both of them. Very good. Restriction endonucleases and DNA ligase. Who gave the answer? Please tell me your name.
I'm Shikhar.
Shikhar, very good, bache. Now, what is the principle behind gel electrophoresis? Right now, I explained. What is the principle behind gel
Both
electrophoresis?
of them.
Both the above.
Yes, yes. Number
Both
one.
of the above.
Yes, very good. Sieving effect of agarose gel. Okay, and DNA being negatively charged move towards positively charged anode. So this is for today. We are going to study gel electrophoretic principle in detail tomorrow. Okay, and we will complete the revision of chapter 11 tomorrow. Okay, so bye bye and thank you. Okay, bye.
Thank you, ma'am.
Thank you.